Hey everyone, I'm Dr. Fizz and let's learn some physiology. Alright, thanks for being here. Today we're going to talk about osmosis, amongst other concepts. Before we get started on our topic of osmosis, it's going to be important that we understand what diffusion is and how it differs from osmosis. Click here to be able to watch my first video and get a better understanding of what diffusion is. Long story short, diffusion is the movement of solute from a high concentration to a low concentration. Today we'll be reviewing osmosis, which is a little different from diffusion. Osmosis is the movement from low particle concentration to high particle concentration. This usually involves a semi-permeable membrane, so we're going to talk about that. First, I'd love to introduce my friend, Mr. Doodle. Mr. Doodle will be helping us out with our drawings for today. Thanks for being here, Mr. Doodle.
But of course,
you always come in handy. Just like diffusion talks about solutes, and we measure solutes through something called molarity, osmosis has a way to be measured as well. In water, we will measure how much stuff or particles is in the solution, and that is called osmolarity. Osmolarity equals particles over liters of solution. Just like molarity measures moles of solute over liters of solvent. We use osmolarity because ions and molecules tend to behave just a little differently in solutions, especially in the body. For example, table salt, NaCl, when in the body, will break down into Na and Cl. Osmolarity is the measurement, the amount of those particles within the solution. Alright, Mr. Doodle, help me out here. Osmolarity is useful when you're trying to describe or compare two different solutions. We have three different classifications for osmolarity. The first one, isoosmotic, meaning that two solutions have the same particle concentration. So Mr. Doodle, please draw for me. Mr. Doodle, please help me out. Show me what two isoosmotic solutions look like. The next one is hyperosmotic. This means that one solution has a higher concentration of particles over the... The next one is hyperosmotic. This means that one solution has a higher concentration of particles over the other solution. And inversely, hypoosmotic means that one solution has a lower concentration than the other. Let's compare how a solution interacts with our cells. This is called tonicity. Both osmolarity and tonicity are essential for understanding how
let's draw it out.
Ions diffuse across our membrane. Tonicity describes how an osmotically different cell interacts with a foreign solution. Where osmolarity separates the two, tonicity is the combination of both of them. Just like osmolarity, tonicity has categories to define states. They are hyperosmotic, hypotonic, and isotonic. The category depends on what you are referring to. Let's look at each of these solutions and compare them to the cell, assuming that solutes can move through the cell. What's super cool is that tonicity and osmolarity can define the same system at the same time. Let's look at an example. Dextrose, or glucose, is an important part of our body's function. Cells use dextrose as energy, and a typical concentration of dextrose in an IV is around 5%, 300 milliosmoles. The same concentration of solute exists within the cell. The normal concentration of saline, which is just salt and water, given to patients is 0.9%, which is the same amount that resides within the cell. When the cell absorbs the solution, the dextrose is metabolizes and disappears within the cell, leaving the saline behind. Say we give a patient a solution of 0.9% saline and 5% dextrose. How would you classify its osmolarity or its tonicity? If you said it was hyperosmotic and isotonic at the same time, you'd be correct. The solution itself has far more particles separated from the cell than the cell does within the cell. So therefore, it would be hyperosmotic towards the cell. And the cell would be hypoosmotic towards the solution. When you combine the cell and the solution together, we would see that the dextrose within the cell is less than the dextrose outside of the cell. And that dextrose and saline, if it was permeable to the membrane, would move into the cell and then be metabolized and disappear, creating another wave of dextrose and saline, or water and salt, moving into the cell repeatedly in order to fix that concentration gradient. If we're not careful, too much of this can actually pop the cells. Generally, the solution would end up being isotonic, because there would be the same amount of saline on the inside and the outside of the cell, and all the dextrose would be absorbed by the cell itself. Now for a brief intermission. Give yourselves a brain break. Okay, so in our first video we established that a cell membrane will typically almost always have a protein that's called a sodium-potassium pump. 2 potassiums are pumped against the concentration gradient into the cell, and 3 sodium ions are pumped against the concentration gradient outside of the cell. This helps to maintain a stable concentration gradient. With different types of channels, letting things in, letting things out, the sodium-potassium pump provides a good equilibrium and allows the net ion charge to be 0 within the cell. The sodium-potassium pump will push potassium into the cell, causing that concentration to increase. When it gets to a certain point, potassium leak channels allow for the exit of these potassium ions. When the potassium ions leave, they take their positive charge with it, thus causing a negative charge to be left in its place. This effectively creates that electrical gradient across the membrane, thus establishing equilibrium. This standard charge on the membrane is called resting membrane potential. Each cells have different resting membrane potentials depending on their amount of channels or what kind of ions they let into the cell and out of the cell. For our standard, we'll look at neurons. Neurons have a resting membrane potential of -70, and we'll use this as an example from here on out. The amount of charge reaches the level where threshold occurs, letting in more sodium, and we skyrocket up to where the Nernst value is for sodium. At that point, the inverse happens for potassium, bringing it back down, repolarizing, and hyperpolarizing, and then reaching resting membrane potential again. That's about it. Thank you guys very much for watching.